welcome to another interesting episode of BioLogic Science News. Today, I bring you a recent discovery made at the University of Texas Health Science Center. The researchers, which include Bruno Doiron, PhD, and Ralph DeFronzo, MD, use a gene transfer technique to get genes into the pancreatic cells of mice with diabetes. As you might know, diabetes is a disease that affects the body's ability to produce insulin and therefore regulate its blood sugar levels. Insulin is a chemical produced by the beta cells in the pancreas, which then go on to enter the bloodstream and break down glucose, which lowers your blood sugar level. In type 1 diabetes, the immune system outright rejects the beta cells and seeks to destroy them. When the immune system destroys all the beta cells, there aren't any cells that can produce insulin. The diabetic can't produce insulin, so they can't regulate their blood sugar level. In type 2 diabetes, the beta cells are defective, and they fail to produce sufficient insulin to adequately regulate your blood sugar. For that matter, what insulin that your body does produce with type 2 diabetes is usually used inefficiently, which means that not only does your body have too little insulin to go around in the first place, what little it does have gets largely wasted. What the researchers did was to take the genes from the beta cells that code for the enzymes that produce insulin, basically the genes responsible for making insulin, and they used a viral transmission vector to get these genes into other pancreatic cells. The researchers reasoned that because these other cell types in the pancreas can exist just as fine in type 1 diabetics, even as the immune system destroys their nearby beta cells, the genes for insulin from the beta cells can be easily transplanted into the nearby cells, and they can take over insulin production without having to worry about being destroyed by the immune system. It's one of those results in science that's both really complex and yet harmoniously simple. After the genes are transplanted, or implanted from a different person, the pancreatic cells begin producing insulin in response to glucose in the blood, just like a healthy beta cell would do. In effect, this simple movement of genes basically co-opted some of the processes of healthy pancreatic cells to fill in for the missing beta cells. Even better, the method doesn't need to be applied to every cell for it to be effective. The researchers say that you only need about 20% or so of the body's capacity to produce insulin to be restored in order for it to sufficiently regulate blood sugar. This is actually really amazing because it's really close to a cure for diabetes. This method could be used to treat and potentially cure type 1 and type 2 diabetes, and if these scientific trials are anything to go by, the risks could be minimal. In the mice used in the study, there were virtually no deleterious side effects. No cramps, no pain, no immune response, no sudden death or glucose misregulation, nothing. But therein is the catch. Despite the really exciting findings, these gene transfers and observations were all done in mice. There would have to be numerous follow-up studies done on human subjects before this treatment could ever come to market. But despite the necessary regulations and testing that needs to be done to ensure human safety and viability of the drug as a pharmaceutical, this study is really good news for those suffering from type 1 or type 2 diabetes. This is a really big step in the right direction towards an effective treatment that might eliminate the need for regular blood testing. Or it could even be a total cure. We don't know. The point is, there's a lot of potential here.